Hello, everybody. Welcome to Talk with Nick. This is the new episode with Karin, who is a postdoc in University of Laval and have done her Ph.D. in University of Quebec. And I've known her for almost three and a half years. And I'm bringing her into this show to know about her journey from Brazil to Canada. And in between, she moved to Australia to do an internship. And she's working with plants and bioinformatics, which is quite interesting for the next 10 to 20 years because it's the booming field. And if you guys are interested in bioinformatics or wants to pursue their higher studies, either in form of PhD, masters or postdoc in Canada or Australia, then this video is for you. So Karen, thank you very much for coming into this show. And
Hello, everybody.
and I'm going to start by asking you the first question. Where are you from and how did you start your studies in bachelor's? What did you choose your major subjects?
Well, I am from Brazil, from a small city in the south, Foz do Iguaçu. A lot of people know it because there's major waterfalls there. And luckily, there was a federal university that opened at my city when I finished my high school. So I was able to start my studies there because before there was no university with public university that offered biology as a bachelor. And when I started, my objective was actually to study zoology. And I did one or two projects in my first year, but I really readily I realized that zoology was not for me. I'm not interested in people. I'm not interested in animals, nothing of that. But in my second year, I tried with a professor in microbiology and plant microbe interactions. And that part interested me. Like, I didn't have to worry about animals really. The microbiology, it was a bit difficult to do this stuff because we didn't have material in the university. So I had to travel a lot to the next country, to Argentina, like 300 kilometers to do any experiment. But it was much more fun than the works in zoology that I tried. So I started doing that. And then yeah, in Brazil, we don't have majors and minors, but at that point, I decided that I wanted to study plants.
All right. And you studied plants for 4 years in your bachelor's. And when did you get introduced to bioinformatics?
Um, so actually, it was a bit weird because when it was my fourth year in, like, I had some courses that introduced bioinformatics, but bioinformatics was, oh, you can do blast. So this was the maximum of bioinformatics, or how to construct a phylogenetic tree. Like, that was the max that we had. But in my fourth year, I got the opportunity to come to Canada for an internship in plant microbe interactions. And it was always my dream to come to Canada. Like, I was 12 years old and I said, oh, I want to go to Canada. I didn't even know really other countries at all, like what other countries were like, but I know I want to go to Canada. So I got this opportunity and I took it. And I said, I want to live there. So my professor said something about, yeah, it's difficult to get students that are interested in bioinformatics. And I'm like, yeah, you know what? I'm interested, but I knew nothing about it. So it was like just to get the opportunity to come to Canada. Like,
Okay.
so I got the opportunity. Well, time to learn it.
Okay. So you when you came for an internship in Canada, was it funded by any particular organization?
Yeah, so the internship was a partnership between a organization in Brazil, CAPS, which is the organization that funds research in Brazil, and an organization here from Canada, MyTex, and they fund a lot of internships of people that come to Canada for to study, or and people that go outside to of Canada. So people study here and go to the to outside to for internships. So I got the like my tax did all the process of selecting the students, but in the end it was Brazil that paid my internship.
Oh, okay. And you spent almost 3 to 4 months in your internship program. And where did you do this internship in Canada?
Yeah, so my internship was of 3 months, 12 weeks. It's a standard for my tax, and it was in Trois-Rivières in Quebec. So like I wanted to come to Canada, and when the professors selected me, I chose the one that was in Quebec because I always wanted to learn French. Like I wasn't fluent yet in English, and I wanted to learn another language. So it was like probably they won't mind that I'm not fluent in English because they probably aren't either because they speak French. So it was like, yeah, I learned the two languages together. So yeah, so I came for 3 months here in well here in Quebec in Trois-Rivières.
So how long it took you to learn the French language? Were you able to become fluent in 12 weeks?
Well, I only when I came, it was a weird year for my professor because there was only one other student in the lab for 2 months, and she only spoke French. Like she spoke a bit English, but it was uh, the bare minimum. So like there was one thing that I joked with her for a long time, that she asked me where was where were the scissors. And I said, oh, they're in the drawer. And I continued doing everything that I was doing. So we were speaking in French, in English. And she went to the where the drawer was, and she stayed there for like 5 minutes. Then she came back, Karen, what is a drawer? Yeah, so it was like we tried speaking English, and I was trying to learn French too. But like I had a few courses in the university, but it was really the bare minimum. Like I could go out and buy stuff in the grocery without an issue. But during the internship, I learned very I didn't learn much. My classes were for like 2 months, but two classes per week, 1 hour and a half, maybe. So I wasn't really learning much while I was here. When I came back to Brazil, when I went back to Brazil, since I knew that I wanted to come back, I started to use Duolingo for the win, because I did all the course. I finished. I got my trophy for finishing the whole course, and then I started watching videos in YouTube to learn the accent from Quebec. And it was so frustrating because I finished the whole course in Duolingo. So I thought, oh yeah, I'm basically fluent. Started watching the videos from French from Quebec. I didn't understand one word. So
Oh my God. Okay, so it's really, really different. Yes. Yeah. Okay,
very, very different.
that's
I
cool.
have a few friends that they learned French from France, like their professors were from France. And even though they are here for 2 3 years, they still have difficulty understanding people speaking French from Quebec. Luckily, because I learned French watching the videos,
Mhm.
I don't have this difficulty. For me, it's weirder the French from France.
Okay. All right, I understand. It's of course weirder when you are learning new things, or you are expressing yourself in different language to somebody else. It's always weird. And I mean, I tried. I'm not very good in languages, and I tried to learn French language for first year, and after that, I gave up because the COVID came up and all those things.
You can't
But
give up.
yeah, Yeah, but this time I gave up because I couldn't like during the COVID time to sit in the Zoom for 3 hours just to learn French, and
Yeah.
then yeah, and then you don't have any hope that you want to stay in Quebec or not. So that was one of the reason. Anyway, then after you completed your bachelor's and your 12 weeks of internships in your bachelor degree, did you do your master's or you directly went for PhD?
So when I went back to Brazil after the internship, before coming going back to Brazil, my professor offered me to come back for the masters with a condition. Said you come back for 1 year of masters and then you do your fast track to the PhD, because I don't have the money to maintain a student for 2 years of masters. Because in Quebec, for foreign students, the tuition fee is extremely expensive. So at the University of Quebec in Trois-Rivières, it's well, when I was a master student, it was $16,000 per year
Oh,
of tuition fee. And well, this includes $1,000 of the health insurance, but it's negligible compared to the rest. this this is just basically, it's just the fee of being an international student. It's basically in the name of the tuition fee for international students. So the the professor couldn't pay me like $30,000 per year to maintain me as a master's student, so I could pay the tuition fee and live normally. So he offered me the scholarship with the condition of doing the fast track. So when I went back to Brazil, I already knew that I would do the fast track. So in 2016, I finished my studies, my bachelor, and then I went back. I came back to Canada for my master's, did 1 year, and then I did the fast track.
okay. And during that 1 year of your master's, did you learn a lot of bioinformatics? And how did you learn the basic skill sets of bioinformatics? Or what kind of knowledge did you gather, and how did you gather it?
Well, I tried learning in the beginning with Data Camp and these other resources online, are mostly, but it was too abstract for me. Like there, I didn't have any data to analyze or anything to play with or anybody to help me indicate what to look for. So I tried and tried for like 2 months and things wouldn't stick in my mind. So I stopped trying there. I did my experiments and then I got the opportunity to go to France for 2 weeks for an intense course in R. The course was R basics because there were other people there learning for ecology, for a lot of stuff. So I went there to just learn the basics in R, and then when I finished the week of the course, I continued another week analyzing data from other people um, in bioinformatics. So I had what we call read counts. So it's just what we get after the sequencing of samples. So I analyzed read counts basically in R. So it was 2 weeks that I was working with data from other people. And when I came back to Canada, my data that I had extracted and sequenced during the holidays, because it was early 2017. When I came back to Canada, I started analyzing my own data. And it was that, like I was learning as I was playing along with my data. So every 2 weeks I would send my professor, oh, here are the results, here are the new results, because I was playing along, and like every 2 weeks my graphs changed because I learned something new that I didn't do in the previous trial.
Yeah.
So it was trial and error.
So if you have to advise somebody to a beginner level student who wants to study bioinformatics, then what would you say? Where should that particular student start? Should he start with R language, or should he start with specifically when we talk about biological data sets, or should he start by understanding biological data sets?
Um, well, that's a difficult question, mainly because I'm biased to R, because I started learning R, so I have difficulty uh, learning in depth Python, for example. Um, but first, I would try to learn the what are the data sets that you want to work with,
Mhm.
and second, learn Unix,
Okay, Unix.
because that's something that you will use no matter what. So this language is the most important. And then depends on what you will work with. For example, if you're working with machine learning in biology, um, a lot of the tools to work with that are in Python. There are tools to work with machine learning in R, but they are better developed for Python.
All right.
So it basically depends on what you want to do. But the other thing that it's good to learn, like the three bases of what you need to learn for bioinformatics are what type of data you will use. So are you going to work with images, with sequencing data, with metagenomics? Then learn Unix, because this is the basis for working in servers, and learn statistics.
Okay.
Statistics are the basis of everything in biology. So I'd say these are the three basic stuff to learn.
Okay.
And a lot of the languages, they are written like in English. So all the commands, if you know English, you will understand what the command is trying to say. So for example, in R, normally the commands will be written in English. For example, mean. So if you know what mean means in English, you will know what the command is trying to do.
All right. That's a good one. So if a student wants to learn bioinformatics, he needs to start playing with R, Unix, and statistics. And is there any particular
R or
reference that
Python,
you?
R or
Okay,
Python.
R or Python.
Because
Okay.
you can do, for example, blast and other types of analysis in Python that you cannot do in R, for example. Unix,
All right.
you can also do blast, for example. And like I have a friend that did hundreds of blast one by one in the web page. These you can do in one line of script,
Okay.
and you
Exactly.
can do the whole set of 100 blasts. So learn Unix, learn Python or R, and you're good to go.
Oh, that's great. So I think students have got the idea that if they want to go into bioinformatics or data science, then these are the basics. And I will pick up on these points later on in the second part of my video, but I will just continue with your journey. Like after you completed your 2 weeks in France and you started playing around with the R language and all these things, you came back, you worked along with your data sets. And I know that you went to Australia for a couple of months to do an internship. So what did you do in Australia, and who funded that trip again? Like who funded that your research stay for you?
Yeah, so my trip to Australia was after I finished my first year of PhD, after my fast track, and it was funded by both the university. I'm trying to think if there was something else that paid my trip. No, I think it was only the university that funded the trip. So the university has a program, of internships outside. So you can either do courses or go for internship, and they pay like $250 per week. So I was really okay to be only with that funding for the 2 months. And I went there to the Australian National University at the research school of biology, and I worked with at the lab of the professor Peter Solomon. I was growing some. Well, I looked into screening some isolates of a fungus if to see if they had a gene of virulence. So basically, if that fungus has that specific gene, it is able to infect and cause, I think it was wheat to die.
Mhm.
So I was screening for that gene and by PCR. And when I found the isolates that I wanted to work, were 10 isolates. Then I started doing sequencing, genome sequencing of those isolates using Minion technology. So this is the Minion is like a USB. It's like this size, and so the library is also like this, and you simply put like there's a lot of work going up to the library, and there's a lot of things that can go wrong preparing the library. But once that is done, you simply put the liquid in the chip, and it sequences at the moment, like so you can use that in the field, for example. I don't know how you would do the DNA extraction in the field, but in theory you can do the sequencing in the field if you need it.
All right.
I know that there was a lot of Ebola analysis in the field with Minayan. So
Okay.
I know that it is possible to do.
So yeah, well, there might be some um, like the small kits for the DNA extraction. They will have optimized the protocol. I'm pretty sure because since last past 10 to 15 years, the DNA extraction method has been optimized. Before we used to do the manual method. Now we have the kits and we could do the DNA extraction in
Yeah.
very less time as compared. So in future there will be possible that we will do the DNA extraction in the mountain areas or in the middle of the ocean. It's going to be
I
really fun.
don't doubt. I think there are some groups that do the analysis in the middle of the forest, like in the Amazonian forest,
Yeah.
the Minayan sequencing. So I just don't know how it is done because
Okay.
my real issue for like 1 month when I started working with the Minayan technology, I stayed the whole month working in optimizing the DNA extraction because it was like what you have in the Minayan is little pores, so it's enzymes, and they what is detected is that a change of electricity, but you need those spores to be like alive. The enzymes need to be working. And as soon as I put the sample in the chip, one 3/4 of the all the pores would die. So what I get from the analysis was not enough to analyze. So yeah, the difficulty is optimizing that, but now it's much better. So I stayed the 2 months basically doing DNA extraction, optimizing library preparation, sequencing, and then I finally started analyzing the data, which was doing the quality control, removing breeds that were of low quality, and then doing genome assembly. And I finally, by the end, I was able to compare two of my assemblies, and uh, we saw some differences in the alignment of the chromosomes. So basically, that was what I was doing. But for example, working there, what I worked with Unix and R for the whole internship. Uh, the DNA, the genome assembly was in Unix using the Canu assembler, and the analysis comparison of the genomes were with are after doing the aligning one genome with the other with Minimap.
Wow, that's a lot of work in 2 months, isn't it?
Yeah, yeah, it
Yeah.
was like by the end, when I finally optimized the library preparation and everything, I prepared so much data that I wasn't able to analyze that because there wasn't space in the server of the university. And like there, I just produced so much. I assembled 4 genomes, and the other 6, there wasn't space to store them.
So how did you
So,
handle this situation? Well, what did you do about the server situation?
well, it wasn't anything for me to do, so I wasn't able to analyze those data. So I prepared, and they stayed there for the team to continue analyzing. I analyzed the 4 genomes that I was able to assemble.
Oh, okay. And when you started the analyzing your data, it did you have to do any more experiments when you came back to University of Quebec, or that 2 months gave you enough data to finish your PhD?
Well, that internship was not related to my PhD. So I went there, basically was cut off of my PhD. I was writing my first paper, but I was not working on anything else from the PhD. And then when I finished my internship, I went back to came back to Canada and was cut off from the analysis from the internship.
All right. So
Normally, these internships they are related to the PhD, but it was not my case.
okay, so it was a separate from your PhD. So when you came back to Canada, you started your own work. So you had your own samples, you did all your sampling and you extracted DNA or RNA and then you send the sequencing. So when you started your work in Canada, was it different as compared to what you learned in Australia, or was it different like in terms of analyzing
In
the
terms
data or
of,
in terms of preparing the samples?
well, yeah, like what I did in Australia was very different from what I was doing in Canada. So in Canada, I was working with Arabidopsis, a model plant, and I was extracting RNA from the leaves, which is it's an easier type of tissue to work with. Um, and working with RNA with Illumina technology, you don't have to worry with breaking the molecule, the RNA molecule. You just worry about degrading the RNA. So you have to worry of contamination or this kind of stuff, but you don't worry that you are manipulating too much the molecule. But for Minion, what you want are the big molecules. So Minion technology has a lot of error compared to Illumina. Illumina, for example, you have 99% of accuracy. The chance of a base being wrong is of 1%, while the chance of a base being wrong in Minion technology is of 15%, if I'm not wrong. So it's a lot of difference. What it's better for the Minion technology is that the molecules are big, so it's easy to assemble a whole genome because you are working with thousands of bases instead of 100 in the size of Illumina. For RNA sequencing for model organisms, Illumina is perfect. For assembly of a non-model organisms, you go normally with the long molecules. So I was working in Australia with a fungus, not a model organism. The fungus has a lot of melanin, so it is difficult to remove that from the when you're doing the extraction, and you are worried. You can't use, for example, a tissue lyser, which is a machine that does like this with a steel ball inside of the tube. So if you do that with Minayan, your sample isn't worth, because you broke all the molecules in small pieces. With Minayan, you have to go by hand with the I don't remember how you name it, but it's a ceramic pot, and the pilon that you have to do like that with the liquid
Mortar and
nitrogen.
pestle.
Yes. So with Minayon, it's by hand. And like there was one day we didn't use kits either. So I was using extraction with chloroform and isopropyl propyl alcohol, and I dropped one drop of chloroform in the lab, and I was with a migraine for the whole day because of that one drop. So it was a lot of work. So it's completely different of what I was doing in Canada. And I wasn't doing in Canada genome assembly. I was working with RNA sequencing with a model organism. I just had to align to the reference. This is basically easy. There's a lot of techniques to do it, a lot of softwares. There are some that super fast, like 10 minutes and you're done. For the genome assembly, you need the whole data. It takes much longer to do it. Uh, it's much more work because there's no reference, basically. That's the problem.
So
So
Australia prepared you for the.
my work in Australia and in Canada, completely different things.
So Australia prepared you and made you more comfortable with the work that you're gonna do that you did in Canada, right? Because in Australia you did a harder job, but when you came to Canada, you find the job that you were doing was a little bit easy.
Well, when I went to Australia, I had more. I already finished all the analysis of my RNA-seq data. So going to Australia, it was like, okay, so this part of my project is already done. When I went to Australia, I was already starting. I had already sent my samples for analysis of metabolomics. So it was the next part of my PhD. So basically, Australia was closing the chapter of RNA-seq,
Oh, that's nice.
but it
And
was adapting to a different environment, because the thing that I found most difficult in Canada, uh, because of the university that I was that I was learning on my own. After, excluding the internship in France, I didn't have a lot of all my learning of bioinformatics was on my own. Like I had one or two courses in person of one day, one in Python and one in Unix. But except for that, I was learning on my own. And I couldn't ask anyone in the lab or at the university for tips on how I could solve an issue that I was having, because nobody knew. I was the only
yeah.
one doing bioinformatics in the whole university. So this was something that was really difficult for me. But when I went to Australia, the lab was on bioinformatics. So it's a completely different environment. You know that you can just pop up in the someone else's lab and ask for an issue that you're having in your script. So it's something completely different.
All right. So did it took you long to finish your PhD? How long it took you to finish your PhD? Because as you said that you were doing it by yourself, and what would have happened if there was somebody who could guide you through your PhD a little bit in a systematic way?
Well, if I had known what I know today, I I don't know if I would have taken less time to finish my PhD, or if I would have cramped up more things in that. Um, but it would have helped me, uh, the time that I was losing on searching for things, like small things. You look at your script and you don't know why it's not working, so you need somebody else to come and look. Oh yeah, you're missing a comma. Sometimes it's that easy, but you take a lot of time because there's no one that can help you figure out what's happening. So I think that's the thing that was most difficult for me. But I didn't take long to finish my PhD. I completed my 3 years in 2020, and I took two more sessions to finish. So basically, I finished in almost 4 years, 3 years, 8 months. But
Okay.
you have to remember that I had 1 year of masters that were basically my time doing RNA extractions. So I took 1 year doing RNA extractions, all the experiments involving that,
Okay.
and that was from my masters.
Okay. Actually, that's quite smart because you started already your PhD project when you were in your master's, right?
Yeah, exactly, exactly. So it doesn't count in the time, but in the work, it's already done.
Yeah. And it takes a lot of time to do the RNA work. Would you say that?
Well, it took me a lot of time because I'm a lot. Well, I was first learning how to work with the things, and um, I I had a lot of a huge issue with my tubes exploding. So this is
Okay.
an issue that you have when working with liquid nitrogen. If one drop of liquid nitrogen falls in your tube and you close without the liquid nitrogen evaporating, the tube will
Mhm.
explode. Use your goggles. That's something that I was always afraid when I was closing my tubes. But RNA, it's so easy to degrade that, and you only find out if it is good quality or not at the end of your extraction. So,
Yeah.
like I did my the first extractions in 2016, and then I was waiting for results of the sequencing and starting analyzing. So I basically find out how many samples I needed in May, and then in May, from May to August, I did all my RNA extractions. I had an intern that I was still joke about it every time we talk about doing RNA extractions. I scream at her, "Don't breathe in my samples," because it's that easy to uh, throw
Really.
all your work to the trash with RNA.
All right, all right. I'm gonna keep this thing in my mind because I have to do a lot of RNA extraction. I have 12 samples, not too many. So I have to do the RNA extraction and send them for sequencing as soon as possible.
Yeah,
Yeah.
so you don't breathe, you don't let anyone move too fast. So and it's alcohol and RNA, RNAs. How do you call it? It's a solution that removes
RNases, yeah.
RNAs.
Yeah.
So it's that all the time.
Yeah. Okay. I'm gonna keep that thing in the mind. So I have done one time and I didn't get one or two times. I didn't get the quality and stuff like this. So I'm gonna keep this thing. Maybe I was talking a lot while doing that thing. All right.
Yeah.
So once you finished your PhD, what was in your mind? Like, how would you use your skill sets to go further? Would you go to an industry or would you go for an academics? And in general, who needs the bioinformaticians in today's world? Like, does it will academics in academic universities or is the industry who's going to hire? I mean, like, where is the opportunities for bioinformaticians?
Well, I don't know if I am really good to answer that, because there are a lot of groups contracting bioinformatician, both in academy and outside in industries. There are a lot of people of industries searching for people that knows how to work with data science and machine learning. Both in Canada and outside, I sent a few applications to one in Australia and one in Germany. Both called me for interviews, but at that time, I had already found a job here in Canada, and my partner lives here, so I said, no, 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 I'm staying. um, but yeah, so you can find opportunities both in the industry and at the academy, and yeah, in my case, I tried applying to both, but it's a weird for me it was a weird position to be in, because as I said, the university where I was, they didn't have a program in bioinformatics. I was the only one doing bioinformatics when I started, so I didn't feel completely confident in applying to jobs as a bioinformatician in the industry. And I didn't find, and to apply to jobs as a plant biologist, I didn't feel comfortable either, because most of my work was not with plants. My professor still jokes with me that every plant I touch dies. So
Okay.
so it was a weird place to be in. So I applied to a lot of places, trying to keep that off my mind, because the imposter syndrome is real.
Mhm.
Um, but yeah, you have to try to keep that off your mind. There is possibilities everywhere. And there's a lot of places of hospitals contracting bioinformaticians. So if you
Yeah.
have worked with cancer data, for example, there are more possibilities.
Yeah, because right now my sister is working in a hospital, and she said like they are doing the lot of single cell sequencing for precision medicine, and now they are investing a lot in the data analysis, and they are moving towards new kind of technologies so that they can give a personalized treatment to an individual, not like a group treatment. So it's really cool that the use of bioinformatics is already happening in different diseases. Hospitals are accepting it. It's really nice. And now I will. Now it's great that you have moved to finish your PhD. You have started your postdoc. It's going to be a new journey, and you will explore different aspects of bioinformatics again. But I would like to move towards the questions about in general about life in Canada, or I would also want to ask you something about life in Australia. Like you had the opportunity to stay in Australia for a very brief period of time, and you have stayed in Canada. So which one is really good for you as you're coming from Brazil, like in terms of lifestyle, in terms of temperature, in terms of also as being an immigrant and living in an international country? Which one would you say is kind of best, and why?
Well, first, being in Quebec is, I think it's different than being in other provinces in Canada because of the language. Most of the other provinces, they speak English. And Quebec, now the policies about immigration are changing. They are being more difficult with immigration. So um, I don't know if it is uh, comfortable for immigration if you want to stay in Quebec, in Canada. Quebec seems to not be as good for that. But before these changes, I felt Canada and Quebec really, really good for being an immigrant. I didn't feel much racism or xenophobia. I didn't feel these types of issues that in Brazil are very, very clear, very obvious. Australia, um, most of my colleagues at the lab, they were from Asia, and I didn't feel that we were excluded, and the students from Asia, they also included me in a lot of activities. So I felt that for being from outside was not an issue. And
Okay.
well, it may be that that was the case because it was a city with a big university, so it's normal to accept people from outside because there's a lot of international students. Maybe smaller cities are not that accepting. For example, you can see that in Montreal, that uh, people don't bat an eye if you are speaking in Spanish in the street. But in Trois-Rivières, that is a smaller city, most people only speak French. If you're speaking in Spanish, people will stop you in the street and, oh, you speak Spanish? Where are you from? So like, I'm having a conversation here.
In a random street with a random person, huh?
Yeah, like um so for being international, a foreigner, being in a small city isn't doesn't feel much comfortable because you feel that all eyes are on you because you're not from there. Here in Canada, I don't know in Australia because I was only at the capital in Canberra. um, but yeah, apart from that, what was the other question?
Like about which one would you choose, being in Australia or being in Canada? Like,
Oh, it depends on what you prefer. Me, I love the cold. I love the snow. So Canada for me is perfect. Australia doesn't have a lot of snow. I was there at the end of the winter, beginning of spring. Um, and like it was -5, the minimum that it reached. And I was going from home to the university on bike, so it's different. The climate's completely different. I come from Brazil. My city is extremely warm, like reaches 50 degrees, but some days in the winter it also reaches -2. So I prefer Quebec because there are some long months of cold.
yeah. So, are you planning to settle down in Quebec?
Um, yeah, I think so. It depends on how the immigration stays, but if there's a lot of problems with immigrating to Quebec, I plan going on another province, maybe Ontario here in Canada, and maybe afterwards I come and settle back in Quebec after having the permanent residency, but after a while living in another province if they don't let me stay well in Quebec. But for now, I really like here because I have the I speak both languages, English and French, and I can interact with people from both groups. And here in Quebec City, a lot of people speak. There's a lot of international students that speak French too, Spanish too. I found some that also are from Brazil, so I feel that there's a lot of cultures. So I really
Mhm.
like that here. I don't know that in from other places, other provinces because I haven't visited. But yeah,
Mhm.
for now, my plan is on staying in Quebec.
That's really nice. Okay. Well, you are clear about it. That's the good thing. And my next question is about the stipend in Canada in PhD and postdoc. And if you have any idea about the stipends in any other country, and if you can compare how is a how is a salary for a PhD student in Canada or being a postdoc. So is it good?
Well, normally in Canada, the scholarships or professors paying students for PhD, it goes from $18,000 to 25. There's a incredible scholarship, the Vanier scholarship, that is $50,000 per year, but that's like an exception. There's one
Yeah.
in 1 million students that are able to get that scholarship, but normally it's from 18 to 25,000 a year. And as a foreigner, you have to pay extra
The tuition
for
fees, or
the tuition fee. But for the PhD is normally the same as people that are from here, except that you have to pay also for the health insurance. So depends on where you live, it's enough. For example, if you live in Trois Rivières, it's a very small city, things are cheap, rent is very cheap. So I lived in a place with a bedroom, a kitchen, and a bathroom, and I was paying $420. And that was with all included. I didn't have to buy the furniture. I didn't have to pay for electricity, internet, none of those things. And so $420 per month is extremely cheap. When you compare, for example, with Montreal, if you want to live alone, that 420 doesn't pay even for a studio.
yeah,
In
Montreal
some
is really
places you have
expensive.
to live very, very far from the university, or you have to share with other people.
Okay,
So yeah, the stipend for PhD, it's okay if you're living in a small cheap city.
okay. And what about postdoc?
Well, as a postdoc, you already have to pay taxes. It's as a student, you don't pay any taxes. So you say $18,000. That's what you're receiving for the whole year. As a postdoc, I receive um, 30, 37,000, but I have to pay taxes over that. So in the end, what I'm actually receiving is more close to $26,000 per year. And then you have to pay, like normally small cities won't have the opportunities for postdoc. So you have to pay, to go for bigger cities, which are more expensive. So then everything gets more expensive, food, the rent, everything else. So it's okay to live. Well, there's a lot of families that live with much less than that and have to pay for to cover kids and everything. So 26,000 dollars per year, it's more than enough. But yeah, you have to account for those differences in the cost of the things.
Yeah. Well, but is it worth it? Like to spend like 4.5 years, strong, very, very difficult 4 years of PhD life, and then going to postdoc with a salary like that. If you go into an industry, maybe you will have 2 times or 3 times of what you're getting as a postdoc, or not as an industrial postdoc, or being an industrial researcher.
It depends on what you want. So if you want to be a professor, for example, you have to go through a postdoc. This is not the case for me, but I want to. My dream job would be being a research assistant as a bioinformatician. So I wouldn't do any of the bureaucracy in the lab, like the professors that are only signing the payment for students, writing grant proposals, doing a lot of bureaucracy and meeting people. No, I would only be helping students with their work. That's my dream job. But I decided to take the postdoc because I wanted to improve my abilities in bioinformatics, because I only learned a small thing in the PhD. It was RNA-seq, RNA differential expression analysis, transcriptome annotation, and the assembly that I learned a bit in Australia. So I wanted to learn to expand a bit my scope before trying to go to the industry.
Well, very well
But
said.
yeah, if you want to expand your scope, it's easier to find something as a postdoc. But if you are able to get a degree in a university that's strong in the field, for example, McGill has a master's and I think doctorate in bioinformatics. So that in that case, you probably will have more support to learn the all the tools needed as a bioinformatician in the industry. Then maybe you are able to go directly to that, and then the pay will be much nicer.
All right. So I just wanted to know because there were many postdoc students or many PhD students asking this question about getting a salary. How much salary is it? Is it enough? Is it enough for a family and things like that? So I think you have answered the questions and bring a few examples, real life, as well as have given other examples too. So it's good.
Yeah, so I think as in the industry, normally bioinformaticians can get from 55 to maybe $75,000 a year, and I think it can support a family. Yeah.
All right. That's great.
As a postdoc, it may be more difficult, mainly if you are the only one working in the family. But I have friends working as postdocs and they have a family, so the salary is enough.
All right. That's great. So I think if somebody's watching, they've got the answer that you can come and it's enough and it's good to start your journey in an international country. Yeah.
For
Okay.
sure.
So I think it's a very good profile, very good journey. You have traveled a lot and you have shared about your troubles in during the PhD, your journey in Australia. Very, very beautiful journey. And I would like to take this conversation to a general aspect of bioinformatics. Like if there is a 18-year-old kid and 18 to 22-year-old who is in bachelor's or master's and he wants to explore bioinformatics, because I remember when I was 18-year-old, there was a boom in biotechnology. Like people were saying biotechnology is the new science and stuff like this. And now I see or I hear that bioinformatics is the new stuff. So how would you say something about this kind of thing or this kind of vibe that is going among the students?
Well, I think if saying bioinformatics is the future is a bit, I wouldn't say the bioinformatics is the future or the thing that is booming, but more computational biology. So computational biology includes much more than
Hmm.
bioinformatics. So computational biology, you also analyze images, for example,
Hmm.
and so includes much more. So if you are able to learn data science, machine learning, scripting in a few languages. So you take the basics, Unix, you take something that is more common like Python or R, and you learn these kind of languages, and you focus on the type of analysis and tools that are common in what you want to do. For example, if it is sequencing, or if it is data analysis, or if it is image analysis. So you try to focus on those kind of stuff. There are a lot of jobs for those. There's a lot of fields in medicine working with data, with image analysis, for example, MRI analysis,
Hmm.
um, that you need a lot of computational biology. There are hospitals searching for bioinformaticians, and there is a lot of work in ecology and molecular ecology for bioinformaticians too. So yeah, it is a field that is booming now, and more to be in biology, to do biology, you need to know things in statistics and scripting, computing,
Okay.
informatics in general.
Informatics. Okay. And I have one general question. Do you think that there needs to be a strong or a very systematic collaboration between a bioinformatician and a white biologist? I think that is kind of missing, or not missing, but I don't see. there is a, like among the students, I don't see that. I mean, I never see that the wet biologist is talking to a bioinformatician or understanding what kind of problems are there in the data science, or a wet biologist student does not try to understand the bioinformatician problem, and a bioinformatician does not try to understand the problem of a wet biologist. So this is kind of a thing that I
Yeah,
felt during my PhD, and I'm still like kind of think these kind of questions pop up in my mind.
yeah, I think it is something necessary, because to be able to do a good analysis, a good bioinformatic analysis, you need to know what exactly was done in the lab before the data was numbers. And so you need there need to be a very good talk between the both biologists, and the wet lab biologists need to understand a lot of statistics to know how to design their job
Mhm.
before giving the data to the bioinformatician and say, give me results. No, it's not a you're not able to do that if your design wasn't proper. So that needs to be really clear for the wet lab. And for the bioinformatician, if you don't understand what the goal is from the wet lab, you you're not able to really give the good results. So for example, I saw a lot of analysis of differential expression from a company with some data, but the results were generic because they weren't focused on what the wet lab people wanted. So if they knew what the lab people wanted, they would be able to focus the results on what was of interest. So
Yeah.
I can give you a list of 2,000 genes that were differentially expressed, but maybe the ones that were important were only two, and then you're not even able to read a table or look into a graph because there's too many data. So it needs there needs to be a very clear conversation between the two. This is why I like to do my own analysis of my data, because I knew what I was looking for.
Yeah. Okay. It's a good one. So there is a gap between a biologist and a bioinformatician, and we need to build a bridge onto that so that there could be a better publications and better output from the research work,
Yeah,
right?
I think this gap is still, and mainly because normally the wet lab people will hire a company to do analysis. So the
Yeah.
company will just do the generics if you don't say exactly what you want.
Yeah.
So
So, and I think the wet biologists, the students who are studying wet biology, they are a little bit scared of the big data sets and working with the coding and things like that. So it kind of get messy and they just skip it. But if they don't do it, I think if we or the general wet biologist doesn't explore this area, it's gonna be a little bit more confusing. And I think so. Like so, that's why I'm trying to do one of my objective of my PhD into bioinformatics. So I just get to know how things work in that area. So I'll let's see how it's gonna go. Anyway, and my last question will be like, how do you see this bioinformatics subject evolving in next 5 to 10 years, or maybe let's say 15 years? What kind of changes are gonna happen? Because there will be some changes. There will be some new technologies like artificial intelligence, machine learning, in terms of storing the data, in terms of sampling the data, in terms of extracting the data. There will be many changes in next 15 years. So what kind of changes can we see? And uh, yeah,
I think storing the data right now is one of the things that is most difficult, because the easier we things get in producing the samples. For example, you said in the beginning that extracting the DNA before was manually, but now we have a lot of kits, so you can extract much more samples like that. And the more you produce, in the end, you have so much data that you don't know what to do with that. Um, right now, it's one of the difficulties, how to
Store the
store,
data.
how to analyze in time, uh, the time. There are more and more softwares that are being developed to optimize the time taken to analyze something. So uh, in the beginning, the genomes were the assembly of a genome was a whole publication. It took 10 years. Now you do that in a few hours. And if you don't find something of extremely interesting, it isn't publication worthy because
Mhm.
there's too many genomes out there. Um, so I had something in mind,
Yeah, no problem.
but there's so many things that can change. So there's the s- data storage, the time of analysis. That is a lot of softwares being developed for that, and the most difficult part is visualization. The more data you have, the more difficult it gets to find how to show your data.
Uh,
You're the one doing the analysis. You find everything of interest, but then how do you show that? that's
yeah,
that's something that is very difficult to do, and that's something that it's from scientists, from one science to the next, it will change. um, but there's a lot of in terms of technologies. I think it depends on the field. So as you said, there is single cell sequencing, there is the long molecule sequencing, Minion and PacBio, for example. um, there's also uh, I had something else in mind. My mind goes all over the place. I can't pinpoint what I'm thinking.
that's a
But yeah,
sign of a PhD and doing a PhD and a postdoc. It's everywhere. It's good.
so there's also, if you look outside of the technologies, there
Yeah,
are fields that are growing. So molecular ecology is something that is growing in the last few years. So
it's really cool.
metagenomics,
Yeah.
studying the microbiome in the field, in the soil, the difference in from tree to tree, for example. So there are whole fields that are getting that are being born because of the new technologies. So I think it is difficult to say what will be of bioinformatics in the next 10 years, because maybe there will be a whole new area just for something that it's a single paper today.
That's very well said. Now I'm more curious what's going to happen in next 10 years because definitely you picked up some points about the microbiome, microbial ecology, and all these things. Also, there is a microbiome in the human gut. So all these things are happening, and this is giving me. I'm also getting popped up with a lot of ideas about these subjects. It's really cool. And Karen, thank you so much. I've got a lot of information about bioinformatics, and I've learned about data science in general, which I would not be able to learn in by reading an article. So it's a great way to interview researchers, postdocs, scientists, and get to know about their head. It's a really cool opportunity for me. So thank you very much for giving me your time.
Thank you for inviting me, Nikunj, and I hope you finish very well your PhD.
Yeah, I hope the same. Yeah.